Hello. Hello everyone. My name is Irshad Ali and my topic is the three models of DNA replication and roll number is 227301. And now definition of the replication. In a molecule's biology, DNA replication is the biological process of the producing two identical replicates of DNA from one origin DNA molecules, and DNA replication occur in a living organism acting as the most essential parts of for the biological inheritance. लाइक सिंपली हम बोलेंगे तो डीएनए रिप्लिकेशन एक ऐसा प्रोसेस है जिसमें डीएनए खुद को ही बनाता है एंड इसके थ्री टाइप्स है डीएनए रेप्लिकेशन के जो कि प्रपोज किया था क्रिक ने फर्स्ट इज द सेमी कंजर्वेटिव एंड सेकेंड इज द कंजर्वेटिव एंड डिस्पेरेसिव फर्स्ट इज द सेमी कंजर्वेटिव the semi-conservative relating to the genetic replication in which double-strand molecule of nucleic acid separate into two single strand, each of the which serve as a template for the formation of complementary strand that together with the template form a complete molecules. Like the सेमी कंजर्वेटिव रिप्लिकेशन होता है इसमें लाइक मोस्टली 50% जो है पेरेंट डीएनए का वो रहता है हाफ एंड 50% जो है डॉटर डीएनए का जो सेपरेशन होता है मीन्स मतलब कि हाफ हाफ ऑफ पेरेंट्स एंड हाफ ऑफ लाइक अ डॉटर जो भी स्ट्रैंड तैयार होता है वो हाफ पेरेंट डीएनए रहता है एंड हाफ डॉटर स्ट्रैंड डीएनए रहता है एंड like this. And second is the conservative. In this model, DNA replication result in one molecule that consists of both origin DNA strand and another molecule that consists of two new strands with exactly the same sequence as the original molecule. Like conservative means इसमें कोई भी disturbance नहीं होता like जो भी replication होता है उसमें जो भी न्यू लाइक डिस्टर्बेंस नहीं होता जो भी डीएनए का जो भी न्यू स्ट्रैंड तैयार होता है वो न्यूली डॉटर स्ट्रैंड बन जाता है एंड लास्ट वन इज डिस्पेरेटिव इन दिस डिस्पेरेटिव मॉडल डीएनए रेप्लिकेशन रिजल्ट in two DNA molecules the mixture of the hybrid of parents and daughter DNA. In this model, each individual strand are the patchwork of original and new DNA. Like डिस्पेरेटिव रिप्लिकेशन में जो भी रिप्लिकेट होएगा इसमें पेरेंट डीएनए एंड डॉटर डीएनए दोनों मिक्स होता है ईच अदर एंड एक्सपेरिमेंटली ये एक्सपेरिमेंटली पता चल लाइक एक्सपेरिमेंट करने के बाद जो डीएनए रेप्लिकेशन होता है वो सेमी कंजर्वेटिव मेथड के थ्रू होता है जिसको लाइक प्रूव किया है मेसल्सन एंड स्टॉल इन 1958, जो कि इन्होंने यूज किया था हैवी आइसोटोप ऑफ नाइट्रोजन N15, and they was use the sanctification method. Thank you, sir. Three models of DNA replication, sir. Mes- Meselson Stahl, sir, in 1958. Sorry, sir.
Yes, yes, sir. Okay, sir. Thank you, sir. नो सर.
नहीं आ रहा है सर.
Yes, sir. Just a second. Good evening, everyone. Sir, is it visible? Okay. Good evening, everyone. My topic of presentation is enzymology of DNA replication. In that, I'm going to talk about helicase, DNA polymerase, and ligase. So I'll start with the small introduction about DNA replication. It is a very important biological process where one DNA can replicate into two new DNA. DNA follows semi-conservative method of replication where the DNA strand is separated and each strand acts as a template for the synthesis of new complementary DNA strand. In the year 1955, Arthur Comberg was first to identify the enzymes necessary for DNA replication. So, now the enzymes which are essential during DNA replication are: first is helicase. It is an enzyme present during DNA replication. The role of helicase is to untwist DNA strand from the origin of replication by breaking the hydrogen bonds of the nitrogenous bases of the two strands and opening them. The energy for unwinding comes from hydrolysis of ATP. There is one more function of helicase, that is, it recruits DNA primase. DNA primase is a modified RNA polymerase whose function is to initiate the synthesis of DNA strand, and the direction in which helicase moves is in 5' to 3' direction. Next important enzyme is DNA polymerase. These are the enzymes that create DNA molecules by assembling nucleotides. They are also called the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from one original DNA molecule. So there are 5 types of DNA polymerase. Let's see one by one with the respective function. First is DNA polymerase 1. It consists of one polypeptide. It is encoded by Pol A gene and it replicates in 5' to 3' direction. It shows exonuclease activity from 3' to 5' direction, that is primer removal activity. It removes the primer when the required DNA synthesis is completed. So therefore DNA polymerase 1 replaces RNA primer with DNA. And it also shows proofreading activity as it can remove any wrong nucleotide which is placed in the 3' end of the DNA chain. Next is DNA polymerase 2. It is a single polypeptide encoded by Pol B gene and its function is in DNA repair. Next is DNA polymerase 3. It consists of 3 polypeptides and its function is to add nucleotides in the uh, DNA strand. So the leading strand needs a single RNA primer for its synthesis, whereas lagging strand needs a series of primers as it synthesizes discontinuously, and each RNA primer is extended by the addition of DNA nucleotides by DNA polymerase 3. And DNA polymerase shows its activity in 5' to 3' direction and shows exonuclease activity in 3' to 5' direction. DNA polymerase 4 and 5 are used in DNA repair. Next enzyme is ligase. The function of this enzyme is very simple. As we all know, DNA primase synthesizes DNA discontinuously in the lagging strand, and short lengths of DNA called Okazaki fragments are formed. When the RNA primer is removed by DNA polymerase 1, there in that place a nick is formed, which it means a small gap is remain. So what does happen is here the DNA ligase comes and seals the gap, joining the fragments into continuous DNA strand. So long lengths of DNA strand is formed, and not only in DNA replication, DNA ligase also is helpful during DNA damage and in the process of recombination. Thank you. Yes, sir. Exonuclease activity is means that it is proofreading activity. Uh, so here what happens is if any wrong nucleotide is added at the 3' end of the DNA chain, so what it does is it removes it. So right uh, right nucleotide could be added there. Difference between the I didn't get you, so okay. Sir, I don't know. Mm, hydrogen bonds.
Yes, sir. Myself, Tanuja Pawar, roll number 5. Today I'm going to present enzymology of DNA replication with the help of enzymes, topoisomerase, SSB proteins, and TUS proteins. Shit. Okay. So, uh, starting by the definition of DNA replication, it is a biological process. As my friend Irshad previously explained, what is DNA replication? So, DNA replication is the process in which DNA divides itself into two uh, for continuing the uh, the the number of cells so that it can uh, It can replace the damaged cells, and it can help in growth of an individual. So this process is carried out with the help of enzymes, proteins, and which play various function during DNA replication. So the first protein here is topoisomerase. Topoisomerase, which is also known as DNA gyrase. It is a nuclear enzyme used in DNA replication during transcription, chromosome segregation, and recombination. Chromosome segregation is the process in which the chromosome splits. I hope everybody knows it was involved in 12th standard during mitosis. And recombination, a class of enzymes that reduce supercoiling in DNA by breaking and rejoining one or both strands of the DNA molecule. This DNA gyrase or topoisomerase 2, it is present at the very start of the primosome complex, which is gyrase, helicase, and uh, and and DNA polymerase 3. Uh, and it relieves the tension so that the helicase can break the DNA bonds, and the DNA bonds jumble, and the replication uh, continue in a can continue smoothly. Catalyzes the ATP dependent negative supercoiling of double stranded closed circular DNA. SSB proteins. The single-stranded DNA of the lagging strand created by the action of the helicase is covered by tightly binding single-stranded DNA binding proteins. When the helicase breaks the bonding between two strands of DNA, both the strands, one of the strand is leading strand, which travel the activity of the polymerase 3 in the leading strand is continuous, and the second one is semi-discontinuous strand, which forms Okazaki fragments. For the till the time DNA polymerase forms the Okazaki fragments on the lagging strand, SSB proteins bind to the lagging strand so that the lagging strand doesn't react with uh, nucleases and other enzymes present in the vicinity. SSB proteins protect the single strand from nucleases until it can be replicated, prevent the formation of secondary structure in the single-stranded DNA that can inhibit polymerases and bind specifically to several other replication proteins, including polymerases and primases. So the main function of DNA gyrase and SSB proteins in short is DNA gyrase assists unwinding and SSB proteins stabilize single strand of DNA. So this is the structure. Uh, the SSB proteins here are the two circular structures. The DNA gyrase is the pink circular structure. DNA gyrase is encoded by GYRA and GYRB gene, that is the subunits of gyrase. SSB proteins are encoded by small SSB gene. Thank you. Enzymology. Actually, sir, I forgot to mention test protein, but I can explain. So, test protein is the protein present at the ending of DNA. It is present on only one side, and what happens when DNA starts replicating? It forms a replication bubble in which the DNA continues to replicate and form form the strands. And when DNA hits this TUS protein, it knows that we should now stop the replication process. So TUS protein is used for stopping the replication of DNA. Also, the gene by which it is encoded is small TUS. Terminal binding protein. Terminal binding protein. TUS. नहीं पता सर. It just releases the tension, nothing else. What? Topoisomerism? Restriction endonuclease forms the nick, and as soon as it forms a nick, the terminal from where the replication is going to start, gyrase goes and binds to that site, and it starts relieving the tension between two DNA strands, which are supercoiled due to due to the compact structure, due to its compact structure. Sure.
Yes, sir. Yes, sir. Sir. Yes, sir. Am I audible? Okay. Good evening, everyone. My name is Savan, role number 06, and my topic for presentation is rolling circle replication. We will start. What is replication? DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms. Introduction. Rolling replication is a process of unidirectional nucleic acid replication can rapidly synthesize multiple copies of circular molecules of DNA or RNA such as plasmids. Some eukaryotic viruses also replicate via rolling circle replication, widely used in molecular biology and biomedical nanotechnology. Steps involved in rolling circle replication are circular double-stranded DNA will be mixed. 3' end is elongated leading strand. 5' end is displaced lagging strand made up of double-stranded Okazaki fragments. Replication of both unmixed and displaced single-stranded DNA. Displaced DNA circulates and synthesizes its own complementary strand. These are the steps involved in rolling circle replication. Rolling circle replication mechanism in DNA. The process of DNA replication is initiated by initiated protein, which makes at the site called the double-stranded origin on one strand of the double strand. The initiated protein remains on the 5' phosphate mixed strand and the 3' hydroxyl end of the mixed strand is elongated by DNA polymerase 3. The unmixed strand acts as the template strand for replication and the 5' phosphate mixed strand is replaced by helicase. Eventually, the mixed strand is completely replaced by newly synthesized strand and will remove itself from the original circular DNA by the same initiator protein looking at the terminating sequence on the mixed strand. The mixed strand then form a new single-stranded circular DNA molecule. RNA polymerase and DNA polymerase 3 then uses the single strand as a template to form new double-stranded circular DNA. Examples in viral DNA, human horse virus, human papilloma virus, Germany virus. And these are the examples in viral DNA. Yeah, that's it. Thank you. Yes, sir. Sorry for your voice. Hello. Yes, 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 sir. Yes, sir. Hmm. Yes, sir. No, no. I am.
Yes, sir.
So,
Yes, sir. Okay, sir. Sir, it is visible. Okay, sir. Hello, everyone. Hello, everyone. Hello. Okay, sir. Hello, everyone. My name is Sakshi Amle. My roll number is 227302. And today I am going to introduction. Matthew Michelson and Franklin Starr in 1958 performed the most beautiful experiment in biology, which supported the hypothesis proposed by Watson and Crick that DNA replication was semi-conservative. This Matthew Michelson and Franklin Starr, they were proposed experiment of DNA replication on E. coli by semi-conservative method. So, what is DNA replication? DNA replication means it is the most to produce new molecules that have the same base sequence, occurs during interphase of the cell cycle, especially in S phase. DNA replication is especially in S phase, and there is the parts of interphase G1, S, G2, and M. So, modes of DNA replication. There are three modes of DNA replication: semi-conservative, conservative, or dispersive. First, semi-conservative. Dotted reflex made up of one parental and only new synthesized strands. Means in this model, the two strand DNA unwind from each other and acts as a template for synthesis of a new complementary strand. This results in two DNA molecules with one origin, with one is new old strand and other is new strand. Then next it is then next conservative. In conservative, original strands stay together after serving as templates for two new strands, and that also stay together. One contains only old DNA and the other only new DNA. And in dispersive, integrate integrity of both parental strands disrupted new duplex strands made up of old and new DNA. Neither the parental strands or nor the parental duplex is present. Means what in dispersive there are strands are mixed, either old, neither the parental or not the parental duplex, and both are mixed, old and new, and disrupted. Now this is the diagrams of types of DNA replication. Semi-conservative model, a conservative model, and the third one is dispersive model. In semi-conservative model, all nucleotides are separated in two DNA. One is into DNA molecules. One is parental or new DNA, and second one is a parental or they are again separated into one DNA gives one parental or new DNA, and second one is both strands are new. And in second one, also first first I given both new strand, and second one is one parental or one new strand.
In conservative model, they are separated in two strands are whole, in second two strands are new, and they are again separated in one both strands are old strands and second one is new strand, and in new nucleotides both DNA strands are new, and in dispersing model old old strands are separated into two in one DNA molecule. Two strands are disrupted mixed when either nor the parental or nor the new strand, and in second also both are mixed strands, parental also and new also. Then let's see Michelson stat experiment. Michelson and Charles conducted their famous experiments on DNA replication using E. coli bacteria as a model system. They began by growing E. coli in a medium or nutrient growth containing a heavy isotope of nitrogen, and then bacteria took up the nitrogen and used it to synthesize three biological molecules into the nucleus. मतलब Michelson और Stan ये जो एक्सपेरिमेंट है ना ये इकोलाइट करके डीएनए का रेप्लिकेशन करने के लिए इसके लिए उन्होंने सबसे पहले हेवी आइसोटोप ऑफ नाइट्रोजन लिया N15 और उसमें डीएनए इसमें इकोलाइट को ग्रो किया and then after many generations growing in the medium, the nitrogenous phase of the bacteria DNA were all labeled with heavy N15. Then the bacteria were switched to medium containing a light N14 isotope and allowed to grow for several generations. Then उन्होंने सेकेंड लाइट मीडियम लिया N14 और उसमें इकोलाई को ग्रो किया. Then DNA made after the switch will have to be made up of 14 as it will have the only nitrogen available for DNA synthesis. Then they collect small samples in each generation and then extract and purify the DNA and measure the density of the DNA using density gradient centrifugation. मतलब उन्होंने दोनों में से भी सैंपल कलेक्ट करके एक्सट्रैक्ट डीएनए को एक्सट्रैक्ट करके प्यूरिफाई किया एंड उसका डेंसिटी मेजर किया by using density gradient centrifugation. Then cesium chloride forms the density gradient from the top to the bottom of the spring. Then here is the diagram of result of the experiment. Here it is shown in E. coli or E. coli DNA. Then they add 15 unlabeled cells to N14 medium and grow for 4 generations. उन्होंने N15 तो N14 में ऐड किया 4 जनरेशन उसमें से 4 जनरेशन शो किए इन जनरेशन 0 उन्होंने उसमें जनरेशन 0 में उन्होंने ओनली हेवी नाइट्रोजन 15 को रखा इसकी वजह से दे कंटेन ओनली दे प्रोड्यूस ओनली सिंगल बैंड उसको खाली सिंगल बैंड प्रोड्यूस किया इसके लिए 100% शो कर रहा है देन इन जनरेशन 1 आफ्टर 20 मिनट्स the intermediate band told Michelson and Stahl that the DNA molecules made in the first round of replication was a hybrid of heavy and light DNA. These results fit with the dispersive and semi-conservative model, but not with the conservative model. मतलब ये इन जनरेशन 1 में जो है वो खाली डिस्पर्सिव एंड सेमी कंजर्वेटिव मॉडल दिखा है बट नॉट अ कंजर्वेटिव मॉडल और उसका जो हैवी नाइट्रोजन जो है पार्शियली लाइट हुआ है एंड इन जनरेशन 2 it produces 2 bands, one with light and second one with heavy. Means 50% light and 50% heavy. And in generation 3 and 4, Over the third and fourth generation, expect the hybrid tank to become progressive painter and the light band to become progressive stronger. Means uh, जो M15 वाला हेवी नाइट्रोजन है वो पार्शियली पेंटर दिखा रहा है और जो M14 वाला जो है वो प्रोग्रेसिवली स्ट्रॉन्गर दिखा रहा है जिसकी वजह से as we can see that the Michelson and Stars are just this pattern and they result confirmed in a semi-conservative replication. The conclusion is Mason and Starr demonstrated that DNA semi-conservatively, meaning that each strand in the DNA molecule serves as a template for a synthesis of a new complementary strand. Yes, sir. Nitrogen, cesium chloride, cesium chloride, cesium chloride, isotope, isotope. So, 2 minutes. Sir, sodium, hello sir, sodium, what after liquid solution. मतलब कौन से जनरेशन के डिस्पर्सिव
Sorry, sir.
ओके थैंक यू सर
आपका यार है सर।
सर नहीं आ रहा है।
Yes, sir.
सर क्या कल सुबह 7 to 8 लेक्चर आपका
सर 1.5 घंटे का लेक्चर रहेगा
ऑनलाइन?
सर ऑनलाइन ना?
सर ऑनलाइन नहीं समझता। ओके सर।
ओके सर, थैंक यू सर।
सर
थैंक यू।
थैंक
सर
यू।
कल कौन
बाय
से रोल
सर।
नंबर का होगा? सर कल कौन से रोल नंबर का होगा प्रेजेंटेशन? ओके सर, थैंक यू सर। थैंक यू सर।
बाय
बाय
सर।
सर, टेक
थैंक
से
यू सर, बाय सर।
गुड नाइट सर, थैंक यू सर।